Okay, well, welcome to another episode of Crop Protection Network TV. Today, we're going to talk about soybean sudden death syndrome and an update on that disease and management. Today, we have Daren Mueller from Iowa State University, Ryan Hamilton, who's a PhD student here at Michigan State, and myself, Marty Chilvers. So let's jump into it, guys. Okay, Ryan, do you want to kick us off?
Yes, absolutely. Thanks, Marty. Soybean Sudden Death Syndrome, or SDS, is an extremely important yield limiting disease of soybeans in the United States and all the major soybean producing regions in the Western Hemisphere. It was first identified in Arkansas in 1971, and since then has spread to all the major soybean producing states in the United States, as well as Ontario and Canada. The primary causal agent of SDS is Fusarium burgiliiforme, at least in North America, but Fusarium brasiliensis also has been detected in certain locations. Average yield losses to SDS total over 932,000 metric tons with an economic value of 276 million US dollars.
Okay, so here's what we know currently about the soybean sudden death syndrome disease cycle. So Ryan's done a really nice job of adapting this figure. So we have spores that can survive in the soil. There's a type of spore called a chlamydia spore that can survive in the soil for many years. It can also survive on plant residue. So we have a bunch of spores in the soil that can potentially germinate and then infect the root system. Infection can occur basically anytime after planting. It does appear that the early infection is kind of important for heavy soybean sudden death syndrome damage. In terms of the infection process, once the pathogen infects, we can have movement of that pathogen further inside of that root section. And once we get movement into the xylem, then we can start having transport of some of those toxins that have been characterized. And that's when we start seeing the classic soybean sudden death syndrome symptoms, the interveinal chlorosis and necrosis. And then, essentially, the disease cycle is going to repeat itself. Spores are going to be produced to carry through to the next year's crops.
So I'll hop in here. Go back to that slide, Marty. One of the things that I always point out is, and it's depicted really well on here, is that like the fungus never leaves the soil. And so there's a lot of, it's really the toxin that, so there's the two phases to the disease. There's that, all of the activity that's in the soil, and then that toxin being produced is almost like a secondary thing and that's getting pushed up to the top of the plant. So I really like your diagram.
Yeah.
Thanks. I think it's important to note too that plant health is seriously diminished by root colonization, even if we don't see foliar symptoms develop that plant is actually suffering.
Yeah, I think that's one of the things that we've really wondered about a lot. When there's that root infection, obviously that's going to have some sort of impact on the plant, but it can be hard to capture that in terms of treatments and yield differences in research trials, at least. So it might be a little bit like soybean cyst nematode, that there may be some hidden yield loss occurring as well. All right. So as Daren was saying, there's clearly two phases to this. There's the root rot element. We have rotted roots. That's not good. And then we have the foliar symptoms that are caused by that toxin. Again, just to reemphasize what Daren said, we only have infection of the roots, but it's the toxins that get moved up to the foliage that result in these symptoms of soybean sudden death syndrome that we see. And just a quick note too, you do have to be very careful. Brown stem rot can produce similar types of symptoms. Red crown rot as well, a new disease for the US, or relatively new, an emerging disease in the US, may be causing similar symptoms too. So if you're not sure, I think it's really important to get a sample into a diagnostic clinic to confirm it really is soybean sudden death syndrome.
Hey, what were you guys' symptoms in Michigan this year? Because in Iowa, the chlorotic spots, they hit us, I don't know, late July, early August. And then it sort of stayed at that stage for a long time this year, and then it sort of progressed later on. But usually the name sudden death, it happens pretty quickly.
Yes.
In Iowa, that this part is where it gets its name. The progression of foliar symptoms. But in Iowa, man, it sort of stuck in that first box over there, that chlorotic spots for a while. I don't know what you saw this year.
Yeah, it was probably somewhat similar. And a note too, I mean, I think in terms of the timing of onset, we normally talk about it kicking in heavily during reproductive stages once we start, late flowering. Yeah, we probably did have, that sort of slower progression this year, perhaps. I know it certainly can vary. Heavy rainfall events tend to drive it. Yeah, it's interesting, Daren. You make note of that difference this year.
Yeah, I would agree with that. At the sites that I was visiting where we know we have high pressure, progress was slower than I expected it to be this year.
Mm.
And which, as a pathologist, was a bit frustrating. I don't know that I actually saw wholesale defoliation of really any plants at any of the sites where I would have expected to see that.
Yeah.
Yeah, I never got to defoliation in Iowa in any of my plots either.
Mm-hmm. But like you noted, it certainly can. That's where it gets its name from. It can turn very, very quickly and go from very healthy looking plants through to defoliation under those optimal disease years, on the disease side of things anyway. Cool. So will IPM work for SDS management? Obviously, the disease is complex that we've sort of been talking about here in terms of onset and symptomology and root infection and toxin production. But certainly, and I think what we'll do here is walk through some of the elements of plant disease management and what may drive disease. So we'll talk about plant resistance, soybean cyst nematode, and cultural practices and seed treatments. So yeah, let's jump into that. So in terms of cultural practices then in SDS, there's been a reasonable amount of work done over the years. And actually, this is one of the studies that was done at Iowa State. And this was something of an accidental study. I really like these slides, Daren. Would you like to walk us through what was done here?
Yeah, you bet. This is the time when Ryan and I like it. Like, we like diseases. And so, having an agronomist wander across the street from their building over to ours and saying, something's going on here. Like there's a problem. And we would walk out there and, these were just some of the photos, but they had some plots that just were devastated with SDS. And it was a long-term rotation study. So it had been several cycles of it. And it's comparing 2 years corn soybean compared to 3 years, which had oats and red clover, and then a 4-year rotation added alfalfa into it. And wherever there was a 2-year rotation, it was absolutely hammered with SDS. And wherever they had a 3 or 4-year rotation, it's just a stepwise. It really did a good job of reducing the amount of SDS in that field. And so that picture on the right really sums it up. This is a same variety, same planting date, same everything, except for you're comparing a 3-year rotation and a 2-year rotation. And the difference is in that photo is just the amount of SDS that's out there.
So Daren, I think one of the questions we get too, in these 3 and 4-year rotations, we've got oat and red clover. So you've got different crop species. When we're talking about rotation from soybean to a couple of years of corn and back to soybean, is that an effective rotation?
Yeah, and so I always tell people that, and this is, you're smarter than me, and so you actually know the science. So I got to say things, and I don't have to back it up. But I tell people that like this pathogen is sort of evolved with the corn soybean rotation. Like, there's a reason why we don't talk about stem canker and brown stem rot and some of the sort of the good old diseases from the 1950s and 60s that were really problematic. This one's emerged right, at the time when corn soybean rotations, or that rotation really tightened down to these 2 crops. And so there's something about corn, like, we've certainly published papers. We're trying to figure out what it is. We don't really have, I don't think we fully know, what the role that corn residue plays, but certainly corn in rotation with soybeans doesn't affect the amount of disease that you end up getting. In fact, sometimes we see a little bit more disease with that corn in the rotation. So I tell people, if you're going to rotate, you rotate to anything but corn and or soybean, and you're probably doing a decent job of breaking up the cycle at some level. But there is a lot we don't know about this whole dynamic still.
Absolutely. And I think there's just some more data there to talk about 2-year versus 3 and 4-year rotations and the amount of SDS over those rotation treatments. They're super interesting. I guess
this is the data that this just shows like it was so dramatic, and it's like, it's almost made up data where you're just like it's a lot of disease and then very little, and then you had a 4th-year rotation actually have even less disease. So
yeah,
it was a clean study.
Yeah, and just back to that corn work again, there's still a lot of questions there. You guys had some folk at Iowa State that maybe found that the Fusarium reguliforme pathogen might be able to colonize corn grain that had fallen onto the ground. And we had a student here at Michigan State that looked at there is some infection of that corn root, but it's nowhere near as great as the amount of like infecting soybeans. So we don't fully understand exactly what's happening here, but certainly rotation away from corn and soybean it can be helpful at reducing disease. And I guess that opens up the element of cover crops too, Daren. So a lot of people are interested in cover crops for soil health and water quality and whatnot. Some more cool Iowa State work. What can you say about some of the work that's happened there and with respect to cover crops?
Right. Well, I think if you look at that previous slide, you don't have to go back, but it's, showing that there are other crops. And when you add them, somehow you get less disease. And so, the logical next question would be, okay, can I intentionally add some of these crops in, not necessarily as a rotation, but as a cover crop? And what would that do? And this is the right question to be asking for any soil-borne pathogen or disease that's out there. And so we've looked at it, across you name whatever disease, and the same for SDS. And so really it could make it worse, which is what we don't want to see, or it could somehow change the soil or change the dynamics that are occurring, and it could possibly make it better. And so the first step was just evaluate a bunch of the plants, and we found that several of them could be infected with Fusarium burgdorferi. So that's the first step. But the next step is like, and this we just looked at rye, but we went out to the field and planted it, and just to see, hey, like, is it making it better or worse, and is it affecting it? And in all of our locations, there was no differences, and so which is good if somebody that's worried about or wanting to adopt cover crops, you certainly don't want to make an existing problem worse.
Yeah, absolutely. Yeah, we still got a lot to learn, but it's great that people are working on this. Fantastic. So there's not been a lot of really recent work, I don't think, on tillage, but there have been some older papers that are out there. What sort of comments do you have with respect to tillage? Can that make it worse, or can it improve soybean sudden death syndrome?
Yeah, I mean, most of the papers that are out there I always call them the knee-jerk reaction papers, where you have a problem or you have a hard pan, and you're going to come in and sort of address that issue that's in that field. And one of the things that we didn't look at, or that wasn't really addressed in those early research papers, was what are the effects of long-term tillage? And we know that a long-term no-till is going to affect bulk density and sort of the soil properties in that field, and that might affect drainage, or might affect some nuance in there that could be affecting SDS. And so we did start a study in 2010 in a field that had a lot of SDS, and we went for about 10 years with the same tillage regime. And essentially what we found, I don't know if it's on the next slide or not, but there are no differences. And so we always tell people, if you're going to till for some kind of disease control, you can, or if you're going to till, do it for a reason that's not disease. And so it's really, we didn't really see any differences. And this is the first couple of years of that. And as that study progressed over the next 5 or 6 years, we had less disease each time we did it, but we also just didn't see any differences.
Yeah, great. Well, that's good to know. Absolutely. Again, what works and what doesn't work. So.
Yep.
Planting date's been an interesting one. It's been an interesting one discussion-wise. I mean, just summarizing here, yeah, we can maybe lower soybean sudden death syndrome by planting late, but do you want to be doing that if you're not maximizing yield? I think is so. the takeaway is, yeah, don't plant late, right? You plant at agronomically optimal times, maximize yield.
Yeah, this is unfortunately, like for white mold and SDS, whenever I talk to people about what are the practices you could do, it's essentially every practice that would lower your yield potential would be a way to manage SDS. And so this slide shows it, the disease bounces around. You do get less root rot for sure, that first phase of the disease when you plant later. The foliar symptoms in this particular case, they bounced around. They weren't really as associated, but I think if you look at it long term, there's 3 or 4 other papers that are out there that show you plant later, you get a little bit, but the risk of getting SDS is lower. But as you plant later, it's really not planting well. You're also managing that yield potential as well and reducing it. So it's a booger. It's unfortunate that the strategies that we could be doing to reduce the risk for SDS, this disease particular is considered a high yield environment disease for a reason. Like everything that you're pushing to get that higher yield is setting up for more SDS.
Absolutely. Well put. So our summary slide, yeah, tillage has minimal effects. That crop rotation away from corn, soybean can be effective. So it's a matter of trying to implement that if possible. And that planting date, we just finish up covering that. So I guess let's keep moving forward then. The other really important interaction we haven't talked about yet is what are the impacts of nematodes with respect to soybean sudden death syndrome. I know there's been work done at various places. We've done a little bit here. You guys have done some as well in other places. But essentially, nematodes can certainly exacerbate soybean sudden death syndrome. And I guess we'll touch on this again in a minute as we talk about risk prediction. Do you have anything to add to that, Daren or Ryan?
Yeah, if it's there, it makes it worse, and it seems to show up a little bit earlier. But yeah, it is a very important factor.
Yeah, absolutely.
Do you think there's a connection between overall reduced plant health due to colonization and damage by the nematodes themselves, or do you think there's other factors that may be at play?
Well, I'll hop in and Marty can correct me, but I think that there is a really interesting dynamic going on, and I don't know if I'll fully answer your question, but one of the papers that was published showed that actually, if you have more SDS, you have less SCN, and then another paper said if you had more SCN, you'd have more SDS, and so it goes back to root health, so if you have really rotted roots from the SDS and that pathogen sort of gets established first, you have less food for the nematode. But if the nematode gets established, what ends up happening is what we think ends up happening is that nematode is actually telling the plant to produce more lateral roots, and the lateral roots are the infection corridor, the infection site for the SDS pathogen. So it's really, again, just like with some of the other things, there's still some PhD dissertations stuck in all of this. As much as we know about SDS, there's still some unknowns in there. So
yeah, certainly. Yeah, I don't think I can really add to that. I agree with what Daren said. So I guess the good news is, though, despite all those challenges, that there are some options. So I mean, obviously, it looks like we need to manage soybean cyst nematode, but plant varieties are really important, and seed treatments can also help to some extent too in managing soybean sudden death syndrome. So let's chat about some of those. So this is a slide that Daren put together here, looking at the amount of total foliar disease and what that sort of means. So we use this abbreviation FDX. It's just a combination of the number of plants infected, so the incidence times the severity. And as we all go through, Ryan's done a lot of this too, we go through rating plots. We'll rate them for the number of plants, so we'll estimate percentage of a plot that has disease, and then we'll estimate of those diseased plants what is the average severity. Is it on a 1 to 9 scale? Multiply those two together to get this FDX component. And there's a very, very clear relationship between the amount of foliar disease and yield loss, with about 0.5% yield reduction for every 1 unit, 1% increase in that FDX score. And you've got some nice slides here of what an FDX of 10 to 20, 25 to 30, and 50 to 60 looks like. So Daren, you've been leading these multi-state studies for quite a few years now. What have we been learning about seed treatments?
Well, let's just establish you've been evaluating them for the same number of years as me. We're equally old here.
Yeah, we are.
But yeah, so I'll say I'm going to reframe it. Like, one of the most common questions I get, I don't know about you, Marty, but somebody will call me up and say, "Hey, I treated my seed with Illivo or Saltro, and I still got SDS." And then they sort of are pointing their fingers, or blame, sort of the blame game is wanting to get started. And I always flip it back right away and say, if you had SDS on that field, you should be happy that you put a seed treatment out there. Yes, you still had some symptoms show up, but without it, you would have more symptoms showing up. So you should thank whoever convinced you to put that seed treatment on your seed, because that field clearly had a high enough risk to get disease. So, and this slide sort of reiterates that. It's a conglomeration. We looked at a big meta-analysis of the first 4 or 5 years of evaluating seed treatments, and this one just has Illivo, but it's just because that's the only product that was available when we were looking at it. And essentially it's showing that it's not eliminating disease. It reduces the foliar symptoms by about 32% on average. Mm-hmm. And if you look at the FDX value, you're getting close to about 20 on average, and that's sort of a typical amount in our research plots. And from all of those trials, we had about a 7% increase in yield. So it's paying off. We did see an increase in yield, but it's certainly not eliminating the disease.
Yeah, I think we're always hoping for those easy solutions. I did this and took care of the problem, but it's very often not how disease management works. And so just understanding that you've got to layer these different elements of management together is really important. So here's some
this is the same idea, but what we did after a couple of years, we like what Ryan mentioned earlier, is that root rot phase is important. We started taking notes on the root rot phase too, and the seed treatments sort of are working. In this particular data set, there's a few less observations. It's only over a couple of years, but we had about 38% less root rot. So there is a component that is protecting that root from the root rot phase. And then this one had a little bit more disease. So we had about 42% reduction in foliar symptoms. So so it's effective against both phases of the disease. Yeah.
And the other thing we do a lot of is testing new products that come through to market as well. And here's a summary, I guess, of 30 site years from 21 to 23. And again, thanks to all the collaborators that participate in that. Daren, so how do we decide on these different treatments that we have listed out here?
I always tell people, and I encourage people, anybody that sees an advertisement for a product that they're claiming to control SDS, or if they have something that they're just excited about on a whim, send it to us. That's sort of our job. And we actually get money from the Soybean Checkoff, and they want us to be evaluating the newest and greatest and shiniest buttons out there. So, that's NCSRP, North Central Soybean Research Program. They give us money to do a product evaluation across the whole Midwest. And so these just be people, either companies or farmers or seed dealers, contacting us and saying, "Hey, can you put in this treatment, or can we look at this combination?" And the particular list that we have here, we actually had 2 or 3 extra treatments that were looked at that were combinations of some of these products, and ones that were experimental that were not going to be available for farmers. So we took those out. But it's literally just advertisements and word of mouth and people asking to include a treatment.
Yeah. And I think that's really valuable work to have that. I think one of the valuable things that we do is work together too as teams and evaluate these products. I don't really want to make recommendations of a single trial in Michigan, but if it's replicated in Iowa and other states, then there's a lot more power in that. So I really appreciate being part of that network and working together. So
And this particular, yeah, these are the results from those 30 locations. And so I'm not going to look at each individual year because there's a lot of numbers here and people can look at it as we sit and talk about it. But if you look at just the averages, those last two columns, you know, the Illevo and Soltro, the fourth and fifth lines down there, they reduce foliar symptoms by 32%. So sort of spot on with what originally we saw back, 7-8 years ago when we first started looking at some of these. And if you look at the other two products, one of them is a combination of several different products, and the other two treatments, they didn't really affect the foliar symptoms that much. That being said, like as always, we could talk about when we get together, like we sit and chat about these things all the time. But there's something really interesting. If you look at the yield for that last treatment, it clearly didn't affect SDS foliar symptoms, but it's protecting yield. There's still a 3 bushel difference between the non-treated control. So there's something going on there. And, and then the scientist in me says, well, there's 4 products there, one of them is contributing, or at least one is contributing. And what exactly is it doing? It's not reducing foliar symptoms of SDS, but it might be affecting the root rot phase, or it might be affecting something else going on. So
yeah, and there may be other factors at locations too. Like we may have a site where we've got high soybean cyst nematode pressure as well. So as we work through these data sets, we need to ask the questions too, like what other diseases are occurring there, what might also be influencing the results that we're looking at. But yeah, very good. Well, I'm glad we do that. It's certainly very helpful to growers to know what's working. So I think one of the things that's really critical for disease management pretty much across every single system is plant variety resistance. And this is a nice photo demonstrating on the left there a variety with susceptibility, and one on the right that's got better tolerance or resistance to disease. So yeah, really important. Part of the challenge there is though that varieties do change out pretty frequently. So I think it's been a bit of a challenge of the university system, like evaluating all these varieties, and we can't always do it because they flip around so quick. We do some evaluations here in Michigan, and Daren, I'm assuming you evaluate some varieties too in Iowa for local companies or national companies. So we do try and work with companies as well and help with this, but it's a challenge, and it's a challenge from the company perspective too, I think, because we're always chasing those high yielding varieties, so they flip over quickly, and so sometimes we don't always get the best rating information on a variety, and that's where problems can occur. And very often where we have those big failures here in Michigan, that's where a variety has been put out and it wasn't really known or understood by the farmer maybe that variety is very susceptible, and then we get caught that, hey, we just from the get-go we should have had a variety with better resistance. So again, just being aware of the problem, which fields have soybean sudden death syndrome and which ones you should pay attention to in terms of variety selection. Yeah. So I think go ahead.
Oh, guys, if I'm not mistaken, there's no resistance to SDS in the germplasm that's available to growers, unlike a disease like Phytophthora, for instance. So the only resistance that's out there is on a continuum of partial resistance.
Yeah, a whole bunch of QTLs, as the breeders talk about, right? There's no major resistance genes that can be incorporated. It's correct. So
yeah. One thing I always tell, like, there's 2 points I try to make, and one is the last bullet point is whenever there's a new herbicide trait that's released, sometimes you don't, those resistance genes don't sort of get incorporated into that first generation of those varieties that are getting released. But like what you said, the last 3 or 4 years, we haven't had the best SDS symptoms. And even in my nursery where I'm inoculating and irrigating and trying to do everything I can to get disease, we don't have the best symptoms. And so companies are trying their best to rate these things, but then every once in a while you'll end up with a 2 or 3 year gap where you don't have good ratings. So then you're sort of basing it off of what QTLs are in there and sort of are they working? And so then a lot of the evaluation happens once the farmer puts that seed out. And so that's when it becomes really important. We're going to end this talk, or near the end of this talk, we're going to be talking about identifying the fields with higher risk. And that's important because the tools that are available, the number one tool is varieties, and sometimes we don't have a good grasp if how effective they're going to be. So
yeah, absolutely. So again, very similar multi-state work here. So again, thanks to all those cooperators, just demonstrating the difference and the dramatic effect of just variety resistance here. So on the left-hand side here, we've got a susceptible variety with a high foliar disease score. That resistant variety planted right next to it, it's not immune. There's basically no varieties that are completely immune to this disease, but much, much lower disease score. And that translates directly into yield. We're looking at, nearly a 10 bushel difference here between that susceptible variety and that one that's got better resistance. So absolutely critical. Any other comments there on variety resistance? Or
Yeah, I mean, just if you look at the percentages, if you remember the seed treatment, it was 32 to 40% reduction. So whenever people are hemming and hawing about where to put their money or their sort of mental energy, I tell them start with a resistant variety. It's more effective than the seed treatment, and it's cheaper as far as extra cost, right?
Yeah.
So
yes, we had a bit of a white mold epidemic in 23 here in Michigan, and I think that's one of the messages I'm trying to get out. It's basically free management. You've just got to select your varieties with reasonable resistance. Obviously, always keeping that yield component in mind, and that's chasing those high yielding varieties. I get that, but we want to make sure that we're also keeping an eye on those disease scores. It's a very, very important component of disease management and risk protection.
Mm-hmm.
All right, talking about risk. Okay, so we would love to be able to receive a soil sample or a GPS coordinate and let you know that, hey, you really should be looking at a resistant variety in a seed treatment in this particular location, or don't worry about it. You got to worry about phytophthora or white mold or whatever it is. So this is some work that we've had going on for the last couple of years now. It's starting to really come through. Daren's got a student and Ryan's a student here working on this. So Daren, what have you guys been looking at on the agronomic side of things?
Yeah, so just to frame it in, we had a lot of locations, and so typically in academics we don't have trials that have something in the hundreds as far as number of locations. So the fact that we have, I mean, this is a testament that we have some of the best colleagues in the world in our little circle, and they're all willing to put out these trials, and so the requirement was have a base seed treatment, a base plus Elevo, and a base plus Saltro. And one or two varieties. If you had two, we wanted one to be susceptible and one to be resistant. And if you had one, it didn't matter. And then in those fields, we all collected a lot of soil samples and then just kept FedEx or UPS or whoever it was happy by, so some of the soil was sent to you and Michigan, some of it was sent to me at Iowa State, and some was sent to Pattern Ag, and some was sent to Midwest Labs. And so we were sending soil everywhere and sort of just getting a really good grasp as to what exactly is going on in the field at the sort of the soil level. And so and then we collected weather data, collected obviously disease data, root rot and foliar symptoms as appropriate, and then yield. And so there was some of it was just computer work too. So this sounds cool and there's a lot of work being done, but a lot of it is sitting at the computer and just downloading the data that we have available. So Ryan can attest to this because he's one of the grad students that has to do the work. You and I just get to talk about it. There's a lot of work that's being done in projects like this.
Absolutely.
And this is just a distribution of the locations, again, all across the Midwest. Down into Arkansas, and then we have a colleague over in Delaware who also put out some locations. And then as we collected data on them, we could sort of fine tune it a little bit. And so some of the locations we knew had higher levels of soybean cyst nematodes. Some of them had higher levels of actual SDS symptoms. And so we could start pulling some of the, as we are doing analyses, we can start pulling some of the locations out and looking at very specific things.
All right. So what did you find from all of this work then? So there's a lot of, we were chatting about what slides to show, how deep to go. There's a whole bunch of complex statistics and data we could really, bring up and bore everyone to death, I guess. But I guess what are the summary from this?
So for the people that like data, they can probably contact us on the side, and we can share more of this stuff. But it really starts to give me a headache just thinking about it a little bit. But ultimately, what we did is we ended up with 4 packages. We try to lump everything that we collected into something that a farmer could manage, something that we could test pretty easily in the soil with just a straight soil test. And then the extra one is the pattern ag. So it'd be sort of a soil test that cost a little bit more. And then the weather variables are something that you can't influence, but certainly you can learn from. And so we sort of broke up all of those factors into these different things. And so I think you can click field, but the category 1 would be farm management, and these would be all of the things that we collected that would be in that farm management one. And then click on the next one.
Oh, yep.
And then category 2 would be SCN populations, soil type, organic matter, et cetera, some of the different nutrient levels. Category 3 is going to be the pattern ag. And so looking at the sort of, and if those are not familiar with pattern ag, it's a company that's looking at DNA profiles and quantifying those. And then, so we sent the soils to them, and then they sent us back the data from these locations. And then the last one's the weather variables, and that's mostly focused on temperature and precipitation, and through either at different times during the year. And it looks like a lot here, but it's essentially trying to figure out when it's important for those rainfalls to occur to get disease.
Yeah.
And then we cut out, okay, fast forward like 10 slides, and there's a whole bunch of stats and different things and different models. And essentially what we did is we took all of those variables, I think there's 51 total, and put them into these 4 regression models. And then at first we tested all of them against each other to make sure that they weren't influencing each other. And we deleted some that were influencing each other. And then we put them into these 4 regression models, ran a bunch of analyses, and then this is actually and then we just ranked them. And so this is actually the values that these are the factors that are coming from the 4 regression models. So again, we cut out about 10 slides of statistics and methods in there. And this is really cool stuff for me. There's things in here that we knew would increase the risk of SDS, like soybean cyst nematode. We've already talked about that. So the fact that it's showing up in here is really cool. I mean, like that's showing like, hey, what, like there's something. It's almost like an acknowledgement of what is being seen, and then also sort of validates what we're doing. Seed treatments is another one, which is, again, we know seed treatments can reduce SDS, and so having that be on the list of things that's sort of adding to the risk of SDS showing up is really important. Mm-hmm. And there's papers published showing pH can it certainly affect it. We like, you know, one of the bullet points for integrated management is having healthy soils, and so we know that phosphorus and potassium and pH, all of those things can influence not just SDS, but can influence SCN, which can influence SDS. And so having that in there. So ultimately, this is not the end game. This is a really nice, clean list. But now the next steps is we're going to go in and start analyzing all of those 123 locations with these things in mind. So looking at, okay, here's the 10 locations that have really high SCN. How did the seed treatments work? And so one of the things that we'll do is pull seed treatments out of the analysis, and then sort of partition it back into these different categories. And so and that's being done. I've seen some drafts of the manuscript. It's getting there, but it's ultimately what we want to do is, can we find fields where we think the seed treatments are going to be more responsive or more needed? So that's ultimately what we're doing with all of these data.
Yeah, it's really cool. I'm really excited about that. Here at Michigan State too, we've been doing some similar sort of risk prediction work. And this is some work we did a couple of years ago now. It was published in 2019. Essentially proof of principle. So we developed a DNA test, what's called a real-time PCR assay, to detect Fusarium virguliforme in the soil. And so were we able to use that to actually predict where soybean sudden death syndrome would develop? And essentially these couple of panels are demonstrating that. So in the soil, we used this qPCR assay to ask how much Fusarium pathogen was in the soil, that Fusarium virguliforme pathogen, and it correlated with the amount of foliar SDS that we found. The other thing that correlated and helped drive things was the amount of soybean cyst nematode. So that's also something that we talk in the spring there. And then, of course, the amount of disease correlates with soybean yield. So we did that across a couple of years. We knew we had a tool we did basically on one farm in a couple of fields, demonstrating proof of principle that we have a tool here that could be used to help predict that risk just based on the amount of that pathogen DNA. And that's where Ryan came into the picture and is utilizing those soil samples that Daren talked about. And essentially, this is what we want to have at the end of the day, isn't it, Ryan? Like a tool where we can predict low pressure, where you probably don't need to worry too much about it, or at medium to high pressure, we're finding a lot of the pathogens. So you probably really should be thinking about that variety resistance and the seed treatment. So Ryan, where are you at in this project? If you want to just sort of talk through the process, I guess.
Yeah, like Daren explained, it begins with soil sampling, a lot of soil sampling. We receive them, we extract DNA from them, along with making note of and collecting all of the soil composition, nutrient data. Once we extract that DNA, we run it through qPCR, which essentially the main point of that is to detect and quantify the abundance of Fusarium virguliforme DNA. We integrate that with all the field trial data that we can possibly muster, and then use a lot of fancy maths and statistics for analysis. Pick out what is the most important and maybe set aside things that are less important and going to maybe confound those models that we're trying to generate. And then it would be wonderful at the end of this project if we came up with a diagnostic tool that we could hand off to anyone in any region where they are able to advise management choices for growers. Say, it's going to be worth it for you to protect your yield by selecting a resistant variety and adding on a seed treatment. Or we may say, I think you have low risk here, and you may not necessarily need to invest in that seed treatment, or selecting a resistant variety may be good enough.
Yeah, absolutely. And in integrating with what Daren's group has found too, with respect to pH or, organic matter and all those other components, right? They're sister projects which are feeding in together, which will be really neat. So we're excited to pull those things along. The other thing, Ryan, that you've spent a lot of time on working through is fungicide efficacy, I guess. And we're always a little bit concerned about fungicide resistance. And obviously, we don't want this Fusarium virguliforme pathogen to develop fungicide resistance, because if it does, those seed treatments might not be effective anymore. So do you want to tell us about what you've been doing to ask that question and to check things out?
Yeah, absolutely. So we've spoken a lot about the efficacy of fungicidal seed treatments, and the two that we've been speaking about use the same mode of action. So if you only have one tool in your toolkit, and resistance develops in the pathogen to that tool, that's not a great thing for the long-term durability of your management using that seed treatment. So what we did was we tested a large panel of Fusarium virguliforme isolates collected from many states, a historical group and a contemporary group, and you can see all of the sample size and date ranges here. Additionally, the historical collection were the very same isolates used in the baseline sensitivity study, also conducted in our lab by a predecessor of mine.
Yeah.
Here's a picture of what that testing actually looks like in the lab. So those are known as poison plates, and we used 8 different treatments, including one to make sure that we weren't confounding our results with the solvent used to actually dissolve the technical grade fungicide. So you can see a very broad difference between colony growth at the same time point when exposed to differing concentrations there.
Yeah. So basically, in the lab, you would use you scan the plate in this instance, but you're essentially looking at how much concentration of the fungicide does it take to reduce that growth by 50%, right? And then that'll give you a number like, oh, this isolate has a sensitivity of about 2 parts per million or 2 milligrams per liter.
Yeah, reporting EC50, or like you said, the concentration at with growth is inhibited by 15%, 50%, I'm sorry, is kind of the standard method of reporting fungicide sensitivity in our line of work.
Yeah. And what did you find from that analysis that you've done so far?
In a nutshell, we found that there was no statistically significant differences between those two groups of isolates to fluopyram, which is the active ingredient in one of the seed treatments that are being deployed out there in the field. It's a very small difference, so 0.24, 0.26-ish milligrams per liter, which is exceedingly small relative to the actual amount of fluopyram that's being applied to each seed. So the story in a nutshell is great. We can continue to use those chemical seed treatments that we have for the time being. The study was conducted about 10 years after the baseline study was conducted. So I'd like to stress the importance of timely and regular monitoring of sensitivity too. So we can start to detect those changes before they actually become a problem for management, and before we see resistance develop in those populations in the field.
And this one's focused on fluopyram, which is the active ingredient in Olevo. We didn't get a chance to screen a whole bunch of isolates against Saltro, the other main seed treatment that we see really good efficacy from. That's called pideflumethafen, but you were able to take at least some of the isolates, right? And what did you see when you compared the two?
Well, the isolates I chose were based on their sensitivity to fluopyram to make sure that I got a good representation of the entire spectrum of sensitivity for fluopyram. And what I found that there was a moderate correlation between them, and it is statistically significant. So the sensitivity to fluopyram in any given isolate is connected to the sensitivity to pideflumethafen, which in terms of when you really examine fungicide sensitivity and how that works biologically, that really makes sense that that would be the case, since they share the same mode of action, they share the same target site, even though they're different compounds and have a different molecular structure. It's something to be aware of and take care of. So when we're advised to rotate modes of action to avoid resistance, that's not really an option here. They're the same mode of action, and so care should be taken.
Yeah, absolutely. Good points. Okay, so yeah, that's kind of what we had to present today. And so this is sort of our summary that we can walk our way through here, or I'll let you read. But I guess, Daren, do you have any other comments in terms of thinking about management of SDS? We've really tried to emphasize like the biology, and it's two parts. It's that root rod and that folio component, which can be a little bit confusing for people, but I think we've gone through and explained that today. The importance of that variety resistance and how seed treatments can be used, and our efforts, I guess, in trying to predict risk and make sure these tools are still effective, such as the fungicide sensitivity work we have going on.
Yeah, I think you summarized it pretty well. I like to put out an encouragement to the farmers and to the companies. The products that we've been looking at that are not Saltro and Olevo haven't looked the best as far as reducing SDS consistently, but I don't want that to be a discouragement. I want that to me is one of the most exciting things, is to see some of those new products that are biologicals coming into the market. And I think long term, as far as sustainability and environmental health, et cetera, we really need to continue to look for some of those that are going to help out with in this in the long term. So I don't want that to be a discouragement to the farmers, to keep encouraging companies to keep looking for those, and hopefully we'll continue to be evaluating them. It has been a couple of down years for SDS, and that always makes me nervous, as far as, like I said earlier, as far as having varieties that we know are resistant. But there was a lot of good evaluation that went on this year, at least in Iowa, because we had a lot of SDS that came in pretty late. So it's a complex disease. I think hopefully you noticed several times that we pointed out that there's holes in our knowledge of everything. We acknowledge that we don't know everything about this disease yet, and even though we have a lot of people studying it for a long time. So it's complicated, but it's certainly fun to work on.
Yeah. And with that, we want to thank all of our collaborators. I think we have most of them listed here. Apologies if we missed your name, but we couldn't do it without our fantastic group of collaborators. And thanks again to the checkoff dollars that helped to support a lot of this work, and some of that industry support as well. And with that, there is more information that you can find at the Crop Protection Network. We have a lot of soybean sudden death syndrome articles up there. We have seed treatment, fungicide efficacy guides that are published and reviewed annually. So go and check out those other resources.